beyond 11 hours. Did we agree?
Yes, so we are going to
Go and
go up to
listen for 30
11.
minutes or 20 minutes.
We're going to go up to 11, up to 12:30.
Sorry, Mike, I didn't get you.
I was saying that we are going to go up to 12:30, which is 1 hour 30 minutes.
Thank you for that confirmation. Are we able to see my screen? Thank you for confirming. So we just wait for 2 more minutes, then we can start. Good morning once more. The time is 11. I think we can start. So I just want to make one announcement. I'm sure by now all of us are aware that we're supposed to be back tomorrow. I know that most of us, if not all of us, assuming that all of us are in second year and you're not carrying a first year course as an overload, you don't have any exams. That is to say, if you're going to come back to campus tomorrow, you must be sure of what you're going to be doing, because there'll be no class definitely for you to accommodate others who are taking first year courses but have got an overload, meaning they have to write exams. So for you, what are you supposed to do in this time? Please make sure that you review your first term material. all that coursework that you've done, and including this one, because we know very well our test ones are also coming very soon. And then from there, we also have our test twos. And then we're going to have our exams. So you can imagine the load that you're going to have as well. So while your friends are going to be having exams, equally, you must also feel the need to study as much. And also just an appeal, we are going to go back to our residences. That's assuming that a good number of us are going to be occupying these, hostels, October, whatever they are. Remember, one of the things that made us go back, one of them is that our systems were being blocked. If you ask me, we have an idea of what was blocking our systems. And how did that stuff get there? It was us. So, yes, they have done a good job, they have repaired them. But if we don't change our attitude, it's just a matter of time before we get back the same situation. So, this calls for all of us to have a conscious. Every time you're holding that sanitary towel, you flush it down the toilet. You're creating room for a similar problem in the near future. So, this is just a humble appeal to everyone to say, as we get back, let's use our systems, our toilets, and other facilities so that we don't get back to the same situation. So going back to our previous lecture on regulation of gene expression, we looked at the operons. What you're seeing in the slide is an example of some of the questions that you're going to expect. And I just want to make sure that before I move on from this topic to another topic, we have understood how to navigate such kind of questions. So I'll start from the top, from this one here. Excuse me. What do we understand here? This is the repressor gene, and we know that this repressor gene with a plus, it means that it is active. When it is active, what that means is that it can produce a repressor molecule. The repressor molecule is that molecule that can diffuse in the system and it binds to the operator. Excuse me, sorry. Let me just take some water. Sorry about that. I'm not too sure what happened to my throat. Maybe one of you doesn't want to come back after hearing that UNZA is opening. Someone is, I don't know. Anyway, what was I trying to say here? We have our I gene. The I gene is a repressor gene from our previous lecture. When you see a plus sign, it means that that gene is functional. By functional, meaning that it can produce the repressor molecule, right? Now, that repressor molecule, when it is produced by a repressor gene that is active, where does it bind in the system? It goes to bind on the operator. But then we also understand that from the Monod and from the Jacob and Monod experiment to say the molecule that is produced by the repressor molecule it can diffuse. If it can diffuse into the system, it means that it can bind to either active or passive operator. In this sense, we have got this operator gene. This operator gene, again, we can see there's a plus there. What this means is that the operator is active. By active, it means it can bind the molecule that the repressor gene has produced, which is a repressor in this case. When this molecule is bound to the operator, when the repressor protein is bound to the operator, it means that when RNA polymerase binds a promoter, and of course a promoter must also be functional, it cannot synthesize the LAC-Z, which is a beta galactosidase, even if it is active, because this position is like a roadblock. It can only produce, the RNA polymerase can only synthesize our LAC-Z if the operator is unblocked. And we know how it is unblocked. It is unblocked if in the environment, in this case, if you're looking at the lactose operon, if there is lactose, because the cell itself doesn't want to spend energy synthesizing an enzyme that is not necessary. So that is why this position is blocked. Now let's look at what is going to happen here. So if we have understood what is happening here, we know when we have got I plus, it means it is functional. When you've got I minus, it means that this gene is not functional. And by that you mean it cannot produce a repressor molecule that can bind to the operator. And then we've got the Z plus. The Z is a lac Z, which is a beta galactosidase, isn't it, enzyme? When you've got a Z plus, it means that if the promoter was able to bind the RNA polymerase and the operator is free, we can get our enzyme, the beta galactosidase. If on this position, like Z, like Y, if you see there is a negative sign, it means that that gene is inactive. Inactive means you cannot get a functional product. And because at this point we have looked at gene expression, we know what a functional product is and what non-functional means. But in this regard, if you're going to use symbols such as a plus, minus, sometimes a C, you must also understand what that means. Plus means it is functional. Minus means it is not functional. Now, you've been given the first situation here or condition is that if lactose is not present, then are you going to get your beta galactosidase or not? This is the first condition. So how you go about answering such questions, you're going to go through each one of them step by step. So we'll start from here. So I'm analyzing this gene, this chromosome, and these are the genes on that chromosome. And together, of course, we're calling them lac operants. We know there are other genes there, but in this question, we've only been given this combination. So I'll start by looking at this interpretation here. So here I've been told that there is no lactose in the system, right? If there's no lactose in the system, am I going to get the beta galactosidase? I'll go back to my gene combination. So my understanding here is that I have the repressor molecule, the protein being produced. It will bind to the operator because it is functional, and because of that, I'm not going to get my beta galactosidase. Now, if there is no lactose, we know very well what lactose does. The system can only produce beta galactosidase if there is a need, but when that means I will not be able to get beta galactosidase. Now, what happens if the situation changes? Okay, what's going to happen if the situation has changed? I'm going to move to this next column. If lactose is present, what happens? Now here we have to be very careful. The assumption here is that there's no glucose in the system, because remember, the preferred source of nutrients, or should I say carbon, is glucose. So this question can only make sense if in the environment there is no glucose. Then the bacteria has to look for an alternative source of nutrients, or carbon in this case. So when we say if lactose is present, it means that in this environment, there is no glucose. So then if there's lactose present in the environment and there's no glucose, what's going to happen to this combination of genes? Remember, when there's lactose present in the environment, this lactose is converted to allolactose. Allolactose will come in the system and it's going to bind to the repressor molecule. Once allolactose binds the repressor molecule, which at this point was bound to the operator, the repressor protein or the molecule no longer has the same affinity to bind to the operator. In short, it changes its conformation. The binding site that it utilized to bind the operator changes its conformation, losing its stable affinity to bind the operator. And together, the repressor molecule and the allolactose move away from the operator. At this point, the operator now is free. What that means is that if the promoter is, in this case, if it is not shown, it means that it is function. It means that now the promoter, which is found upstream of the operator, can bind the RNA polymerase and synthesize beta galactosidase, which can be used in the utilization of lactose. This is this scenario here. I'll go to my next situation. So here, I've got two chromosomes or two combinations. Let me just keep it simple. So I have this combination up here. I have the inducer, the repressor molecule is positive, meaning that it is functional. Because it is functional, I'm going to get my repressor molecule. I'll go to the next position. The operator here is functional. Because it's functional, if in this system the repressor molecule has produced the repressor molecule, it can bind to this operator. Okay? Now, if you look at the LAC-Z gene, it is negative, meaning that I can't get a functional product from this combination up there. It is not active. Let me go to the next combination. This is in the same system, by the way, but to analyze them, you analyze them step by step, just by understanding what each one of them is trying to tell you. So when I go to this position, in this chromosome, what you see is that the inducer molecule is not functional. So meaning that this copy of the gene cannot give me a functional gene product. I can't get my repressor molecule. But remember, at this position, you're still going to get a repressor molecule because the other copy of the gene is functional. So while on this chromosome, this copy is not functional, in this system, you're still going to get a repressor molecule given the fact that this position is functional. Let's go to the operator. The operator on this chromosome is function, meaning that if there's going to be a repressor molecule produced, this protein can bind to this operator. Now, the question here is, this repressor protein, can it bind to its own operator? This is cis. Or can it also bind to this operator? This is trans. If this repressor molecule binds to this operator, we say it's acting in cis. But we're saying, can it also bind to this operator? Why am I asking that question? Because remember, on this chromosome, its repressor gene is not function. So then, if anything should bind there, it must be coming from this copy of this gene. So here, from the previous lecture, we said that this is possible because this repressor molecule that is produced is diffusible. Therefore, it can act in trans. It can bind to this operator as long as it is functional. It can also bind to its own copy of the operator. So meaning that this operator is going to have an effect. So this repressor molecule produced here is going to have an effect on both operators. Now, when you go to this position, on this chromosome, the LAC-Z is not function, but the LAC-Z there is function. So meaning that this operator molecule, if it binds to this repressor molecule, if it binds to the operator here, there's really no consequence because the gene on that chromosome is not even function, right? But what happens if this the repressor molecule that's going to be produced here binds to this operator? It's going to have an effect on the gene product of the LAC-Z because the LAC-Z is functional. Now, let's move to the conditions that you've been given. In the first condition here on this column, I'm moving. So what I'm saying by condition is I'm looking at what is here, and then I'm interpreting what I should put here. So I'm assuming this was going to be blank. So there's no lactose in the system. If there's no lactose in the system, what happens here? I'm going to get my repressor protein. This repressor protein will bind to both operators. The repressor protein is only coming from this gene that is active. It is not coming from this gene at this position. When there's no lactose in the system, the operator position is blocked. What this means is that even though RNA polymerase can bind to the promoter, which in this case we're assuming the promoter is here, I'm not going to get any gene product. Now, is a gene product going to come from here? No, because this guy is not functional, this guy is functional. So as long as there's no lactose, this gene product here would not be obtained. That is why here you're going to say there will be no beta galactosidase. What happens when you put lactose in the system, which is now looking at this condition that you've been given here and the information that you should be able to put, assuming this was not even there? How do we get to the answer? So we go back there. If there's lactose in the system, we know that this lactose is going to be converted to allolactose. Okay, it's an isomer of lactose. Now, if allolactose is present, what does it do? It binds to the repressor protein. At this point, the repressor protein was bound there and was also bound here. The allolactose is going to relieve this operator and that operator. So both operators are going to be free. If both operators are going to be free, we're assuming now that the promoter can now synthesize the gene that is downstream. On this chromosome, there's no gene product because this gene is not functional. What about what's going to happen here? If allolactose binds to this operator, the allolactose binds the repressor molecule. and that results in freeing of this operator. When the polymerase binds the promoter, you can now get the synthesis of LAC-Z. That is why here we're saying beta galactosidase is going to be produced. Lastly, let's look at this combination here. We'll start looking at each loci. Let me just say each position at a go. In the next lecture, we're going to be looking at a specific position on a chromosome where you find your gene. It's called a loci. So let me just call it this position here. Let's look at this position on this chromosome. This is chromosome 1, chromosome 2. They carry the same combination of genes, but different functionality. Either it's active or inactive. Inactive, I mean it is mutated, it's not function. When I say active, it can give you a functional product. So let's start with this position here. When the I gene is, the lac I gene is plus, it means it is function, meaning that both copies of this gene are going to give us what? The repressor protein. Now, that repressor protein, what does it do? It's a diffusible molecule or protein. So meaning that when it diffuses, it can bind to this operator. It can also bind to that operator. But let's look at what is happening at the operator region. I'll start with the top one. On this operator, we can clearly see that the operator is not function. It is it's got a defect. That is why it's designated OC. What this means, but then look at the other operator. The other operator is O plus, meaning that it is function, meaning that it's got an ability to bind. It's not giving us any product, but being function in sense is that it can bind the repressor protein. So while this gene can produce a repressor protein, that repressor protein cannot bind to its own promote operator because it is defective, but it can still bind to this operator. In a similar way, this I gene is going to produce the repressor protein, but repressor protein can only bind to its own operator. It can't bind to this one. If it was active, it would have bound, because we're saying that the product of the LAC-I is diffusible and it can act in trans. It can act in cis and also in trans. Let's look at this position. Here on this chromosome, we can see that this gene for the LAC-Z is functional, but that one there is not function. So then what happens when we don't have any lactose? If there's no lactose in the system, the interpretation is that we're going to get functional repressor proteins. These repressor proteins will diffuse in the system. Where are they going to bind? They can't bind here because this is defective, but they can bind to this operator. Now, once this is bound to this operator, it means we can't get, even if it is bound here, we can't get anything because this guy is defective. Now, keep in mind, this operator here it cannot bind the repressor molecule. When the operator binds the repressor molecule, it blocks transcription. Now, if it is defective and it can't bind the repressor molecule, what does it mean? It means that there is nothing holding the polymerase from transcribing and translating the beta galactosidase. So regardless of whether there's lactose or no lactose, you still get your beta galactosidase. Let me just go through that again. What is happening here? In this system, we have an active repressor molecule being produced, but the operator on this gene is defective. When it is defective, it cannot block transcription of any functional gene that is downstream. What that means is that the polymerase will bind, and whether there's lactose or no lactose, this guy here cannot create the roadblock. It will allow for the transcription of this gene. So meaning that you're going to get beta galactosidase when there's no lactose and also when there's lactose. In this system here, this operator region is bound, is blocked by the repressor molecule. But the only challenge here is that even if there's lactose or no lactose, we can't get anything because the gene that is downstream, it could be LAC-Z, LAC-Y, is not functional. And that is why we cannot consider the product on this chromosome. I hope this is clear. I included this one in your previous notes. I don't know how many of us managed to come up with the answer. I'm going to do the first two. These other two, I want someone to attempt. Now, right now they look very squeezed, but I hope you can follow and, be able to, to figure out the answers. So this time around, you have a combination of beta galactosidase and lactose permase, right? Like why? Now, here we're assuming that there's no lactose. Here there's lactose, again, no lactose, no lactose. And you're being asked, if there's no lactose, are you going to get your beta permase? If there's lactose, are you going to get your beta permase? The interpretation of this table is that right now we've got a combination of two genes. You've got the beta galactosidase with these two columns, and then the lactose permase or just permase, these two columns. For each one of them, you've been given two conditions: no lactose in the system plus lactose in the system, no lactose in the system plus lactose in the system. So let's start with the first genotype. The genotype just means the gene combinations, right? In the previous slide, of course, they were arranged in a way that you can easily follow this position to that position, but I hope even in this case, you're still able to follow. So you have the lac I is positive, that means it is functional, and at this position, it is also functional, meaning that in this system, you're going to get the repressor protein. Let's go to the promoter. The promoter is P plus and they are P plus. That means the promoter is function. What does a functional promoter mean? It means that it can bind RNA polymerase. And in transcription, we know what happens afterwards. If RNA polymerase can bind to the promoter and all the transcription machinery is there, we can have we can have transcription taking place and our RNA transcripts. And finally, if all conditions are permissive, we can have translation and we get our gene product. So when you see P plus, it means the promoter can bind RNA polymerase. The O plus means the operator is functional, like I explained. Z, it means the Z plus means that the beta galactosidase is functional. Now here, you can see you don't have the lac Y, the lactose permase. It means that the lac Y here is negative. Maybe for space, you just take it out. But the assumption here is that because you've got two combinations, it means that on this position here for Y, on this gene, it is defective. Let's move to the other one. I plus, P plus, O plus, Z plus means they're all functional. This time around, we also have Y plus, meaning that the lac permase gene is functional. So now, what's going to happen? Let's look at the gene beta galactosidase first, before we go to lactose permase. What happens when there is no lactose in the system? So the understanding here is that we are going to get our repressor molecule from this gene and from that gene there. This repressor protein can bind the operator. And when it binds the operator, I'm not going to be able to get my lac Z either here or there because both operators are what? Function. They can bind the repressor molecule. The polymerase, was it able to bind the promoter? The assumption is it can. But because there's no lactose, this operator region is blocked. So meaning that I don't get any beta galactosidase. When the opposite happens, we have lactose, and we're assuming there is no glucose in this environment. What happens when there's lactose? When there's lactose, the operator region that was blocked by the repressor molecule is going to be unblocked because allolactose can be converted by beta galactosidase itself to allolactose. And allolactose will bind the repressor molecule, thereby unblocking both of these operator positions. Now, when that happens, meaning that I can get my lac Z. Now, this lac Z, the beta galactosidase that you're seeing as plus, where is it coming from? It is coming both from this copy of the gene and that copy of the gene because they're both function. Let me now move on to lactose permase, the lac Y. Now, in a similar way, when there's no lactose, right? When there's no lactose in the system, both operators are functional, meaning they can bind the repressor protein. So meaning I'm not going to get my lac Y. That interpretation is not from this genotype, it is from this other one because this is the only copy that is function. What happens in reverse when you have lactose in the system? When I have lactose in the system, the understanding is I'm going to have allolactose, which has got a high affinity for the repressor protein, in this case, the lac gene or the lac protein, okay? So that the binding of the repressor molecule, the protein, to allolactose frees both operator positions or regions. When these positions are unblocked, the promoter we're saying is functional. I can get my LAC-Y. The LAC-Y that I'm seeing here as a plus, which gene copy or chromosome is it coming from? It's not coming from this one, it's coming from this one because this is the only genotype where we've got a functional gene copy. And I think, well, let's look at the second one. If you look at the second combination, it is I plus, P plus, OC. I'll start with the I position. The I position here means this is a defective gene, so I'm not going to get any functional product. What about the other copy? Here I'm going to get a functional product. The promoter on both chromosomes are functional. What about the operator? The operator here is not functional, meaning it's going to remain free. If polymerase binds to the promoter, I can get my gene copies as long as they're functional. What about this operator region? This operator is functional because it's O+, meaning that if this repressor molecule produces a protein, it can bind to this position and block the transcription of these genes. The Z here is positive, it's functional. On this copy, it is not functional. So meaning that if I'm going to get any beta-galactosidase, it's going to come from this gene copy. What about the LAC-Y? The LAC-Y on this position is defective, meaning that if in the system I'm going to get my PMase, it's going to come from this gene copy. So let's look at a case where we do not have any lactose. Now, when there's no lactose in the system, what does it mean? It means this gene copy is going to produce the repressor molecule. This repressor molecule can bind to its own operator. And when it binds to its own operator, definitely it can block not LAC-Z because LAC-Z is defective, but it can block the transcription of LAC-Y. What about what's happening there? The repressor molecule can only bind to a functional operator. If this operator molecule is defective, it means that it cannot block the transcription of LAC-Z. And that is why even if there's no lactose, this one is going to be under, it's going to be under constitutive transcription. Nothing is there to block it. Okay, so we're going to get beta-galactosidase both when there's no lactose or when there's no lactose because there's no roadblock there. But what about the PMase? If you look at the lactose PMase, we're saying that the operator is going to be blocked in this situation because it is functional. In this regard, it is not going to be blocked. If there are any genes that are active, they'll be transcribed. But remember, we can't get the LAC-Y from here because the LAC-Y is defective. So meaning that the LAC-Y, if it's going to be in the system, it can only come from this gene copy. Because the operator is free, when there's no lactose, you can't get lactose. That is why it is negative. The moment you introduce lactose in the system, you can unblock your operator and get your lactose PMase. This is it about these kind of questions. I want someone to try this one. And you can also try this one. Anyone? I need to move on to the next lecture. I already spent a good 30 minutes. Did we follow?
No, madam.
Okay, normally when people say no, it's
As
a
for me,
problem.
I'm lost, madam.
The only way I can help you is if you can tell me how you're lost, because it's difficult for me. Where do I start from? Are what are you not understanding exactly? I know this kind of questions are confusing. That's very true. I do appreciate that. But the only way I can help you also is if you help me. Maybe what we need to do here is to look at the understanding of what these I, P, and Z things mean. Okay, or maybe is it when you have I plus, P plus, what are you not really understanding? Let's help each other here. Let me go through one
Yeah,
of them.
madam. Like on the part that of these, the IP, the P plus, the O plus, the Z plus, and the OC. So like, what criteria are you using? What simple logic can we use to tell that, okay, in this one there is no lactose, in this one there is a positive lactose, and then if this and this reacts, if react, what sign are we going to get if we're going to have a positive or a negative? What criteria, what simple criteria can we use to understand it is?
Okay, so and you know, this is me thinking I was explaining like the simplest. Now I need to explain even. Okay, what criteria am I using? I'm not too sure. Okay, help me. Should I use this slide or should I use the one before? Which one is easy? These ones are too squeezed, even for my eyes. Should I use this one
The previous one, yes.
or should I use this one?
Yes, the previous
What is
one.
yes? Okay, let me use the previous one.
Yeah.
Okay. Allow me to use this combination because normally they will exist in this form. Okay. So what do we understand
Hi, in this
by
video, we're
the
going to show you
IG?
how to format a
Here,
larger
we're
USB
trying to say
flash drive
that
as
the
FAT32.
IG
So you may have noticed if you try
is producing a protein. And when it produces a protein, how do I know that it's going to produce a protein? Because remember, on top here, you can see there is a plus, isn't it? If I have a plus sign, it means that this gene can produce a protein that that we're referring to as a repressor molecule. Now, I can also look at this position. Remember, the concept here is that this is a situation in a bacteria. Is that this is a genotype, that these are the genes that you are carrying. Okay. Right now, some of us have got very dark hair. What this means is that we have a gene that can give us that color of hair. But remember, all of us, we carry two copies of that color for hair color. So it means that if your hair is very dark, you've got two copies of the gene that give us a trait, darkness. Okay, I hope you're able to follow. If some of us, when you look at our parents, maybe one of your parents is very light, one of your parents is very dark, but you are dark like me. My mother is very light. My father, my sorry, in peace, was dark. But I'm dark. Of course, I've got siblings who are light. But then while I may appear dark, the color that you're seeing as dark has got two gene copies. The darkness that you are seeing and maybe a lighter version. The version that you are seeing maybe is one that is more dominant. So you see it more. It's being revealed more. But that doesn't mean that I only carry genes for darkness. That is why myself, I've got 4 children. One of them is very light, probably because maybe that other gene can also exhibit itself. So when you look at this kind of a scenario, think of it as this bacteria cell carrying two gene copies that can give us the same kind of product. For example, if this is I plus, it gives me product A. And if this position was also I plus, it should also give me product A, which is the same thing. They should all give me a repressor molecule. If this position is O plus, and this position is also O plus, this position is a binding site, meaning that the structure of this binding site and this binding site must bind to the same thing. If at this position we're supposed to bind a protein B, it means this one should also bind the protein B. So meaning that this bacterial cell is carrying two gene copies of each trait. Okay? The similar thing here, we know this Z is a beta galactosidase. Remember in the previous lecture, we looked at those 3 enzymes, isn't it? So now in this situation, you've only been given one of the enzymes, beta galactosidase, which is used in the utilization of lactose. Now, it means that this organism has got two gene copies giving us gene beta galactosidase. And this is important because maybe when one of them is not working, like in this case, you can still get the same gene product. And this is very good. This is where you think life is really, fine-tuned for fitness or just conserved to preserve life. Because there are times when one of the genes is not working. But if the other one is functional, you can still get to live a healthy life. So this is looking at such a system in a bacterial cell because it is easy to interpret and to analyze. So the understanding here first should come from us appreciating to say, when you see two positions of the same thing, these must be giving us the same gene product. But there are times when one of those copies of the gene is sick, defective, okay? If it is defective in the system, if the other gene is okay, this bacteria cell can still regulate the transcription or the utilization of lactose if it is present in the field, in the environment. So I hope that is very clear. Even yourself, if you look at yourself, maybe even your height, some of us are tall like one of our parents, some of us are, like me. So when you look at all those traits, it means that you're always carrying 2 copies of a gene for the same trait, okay, that we're able to see. Now, in this case, in the bacteria, I'll start from this position. At this point, we all understand when I have an I and an I here, this is the same gene, meaning they can give me the same gene product. But then how do I know whether it is that, whether I can get a gene product from this gene copy or from that gene copy? It is to use what you're seeing here as plus or what you're seeing here as minus. If on this gene I have a plus sign, it means that the product from here is going to be possible. That means I'll get transcription. Remember, we are looking at regulation of gene expression. And in the previous lecture, we said that in bacteria, one of the key steps to regulate gene expression is transcription, because you don't want to, each transcription itself is an energy consuming process, not even energy, even just resources. Translation itself, it's an energy, and it needs a lot of stuff. So the bacteria want to invest so much. So the best position to regulate is transcription. So here looking at the process of transcription in as a way to regulate gene expression. So now, if you have your I gene that is function, on this gene copy, it is not functional, meaning that when you analyze this question, you look at both of them at the same time. Not looking at them independently, by the way. The first thing you do is to analyze them together. Okay, so I'll start with this position here. Let's analyze them together. So we are saying I plus. I plus means I'm going to get my gene product, the repressor protein. Let's just call it a protein, repressor protein. This copy here is sick. Like I said, it's defective. Am I going to get my repressor protein? No. I was only going to get it if this was positive. If this is positive, is a product here going to differ from that one? No, they'll be the same thing, and all of them should function the same. But in this situation, this guy here is sick, will not get any repressor protein. So the repressor protein that we get from here, what does it do? Or where does it bind? When it is produced, one of the important things that we're supposed to know is that it is diffusible. Okay? You can imagine if right now where you're sitting, someone walks in with a very strong perfume. Now, that strong perfume, I don't think it's only them who are going to be able to smell it. Even you can tell, oh wow, someone has walked in with a very nice perfume. If it is food, in fact, we're going towards lunch. If your mother or someone is cooking food in the kitchen, that aroma from the kitchen, I'm sure it is reaching where you are. It can diffuse, isn't it? Meaning that it's not restricted to only the walls of the kitchen. So think of what we're saying here as diffusible, in the sense that when this repressor protein is produced, it is diffusible. If it can diffuse, it can bind both to this operator and that operator. So in this case, imagine yourself right now smelling the aroma that is far away in the kitchen, but you're in the bedroom. That aroma is not only confined to the kitchen because it can diffuse. We are saying that the repressor protein is a diffusible molecule, in the sense that it can diffuse in the system and bind to any functional operator. So here, the key thing to remember is that the repressor protein is diffusible, and this repressor protein, if it is diffusible, it can move in the system. Where does it bind? It doesn't bind to the promoter. It doesn't bind to any of your LAC-Z, LAC-Y genes. Its target is to look for a functional operator. And when it finds it, it will bind there. When it binds there, it acts like a roadblock, okay? It's going to sit at this operator, like right now, it can sit here. Why? Because this operator is function. How do we know? There's a positive sign there, right? So this operator there is also function. Where is it going to sit? It's going to sit there. But remember, what is the repressor protein that is bound to this operator didn't come from its own gene product, because this guy here is sick. Like I said, it's defective, right? So because this guy is diffusible, it can bind to both operator regions. Now, When it binds to this operator, what does it mean? It means that it will block transcription of any genes that are downstream this way. If you had like Y likes, all of them will be blocked. Now, I'm going to move to this position here, assuming we've understood how the operator is functioning. The operator is functional. How do we know this? There's a plus, there's a plus. If the operator is a C, sometimes a minus, it means that operator is sick. That's, you know, we don't say sick, we say it's defective, isn't it? I'm just using sick for us to understand that it is not working. It cannot bind the repressor. Even if it is present in that environment, it cannot bind it. How do I know? The plus sign. Let's move to this position there. Now, if you look at this position here, we can start with the first copy of this gene. Okay, this list could be the gene for, let's say, eye color. Okay, the gene for eye color, we can see that on this chromosome here, on this DNA copy here, it is defective. How do you know? It's got a negative sign. So meaning that even though, you know, at some point transcription were to take place, if at all it took place, we can't get any gene product from here, meaning I cannot get any beta galactosidase from this gene. What about this position here? This position here for the LACZ, it is working, it is functional, it's a functional gene copy. What that means is that if at all there was a chance for transcription to take place, it means that this gene copy will give me beta galactosidase. So the first thing is just to understand what is going on there. Now, when we say plus lactose, minus lactose, how does lactose come in this picture? Remember, lactose is coming in as an inducer to induce the transcription of these genes. Lactose is coming in as an inducer. What do you mean by that? The lactose itself is first of all going to be converted in a form that can attract the repressor molecule. The lactose will not bind the repressor molecule. That is now, at this point, we say this position is function, isn't it? So imagine that the repressor molecule is bound there, so meaning it is blocked. Now, lactose itself cannot bind the repressor molecule in its form. It is converted to allolactose, like an isomer of lactose. Okay, this allolactose has a high affinity. It is very much attracted to the repressor molecule that is already at this point sitting on your operator. Maybe I forgot to mention, when the operator is, when the repressor protein is bound to the repressor molecule that was produced, you cannot have transcription. It is like blocked. So in this case, we have a roadblock. How do those, how is a roadblock unblocked? It is only unblocked when in the system you introduce lactose. And in that system, like I said, there should be no glucose, because if there's glucose, the bacteria has got a preference. It will rather use glucose than lactose, and we know why. Glucose is a monosaccharide. Lactose is a disaccharide. Why should you spend more energy breaking down a disaccharide when you've got something simpler to use? The bacteria, you know, has got a very big brain, so it can decide. So now in this case, when you say plus or minus lactose, your reference point to start with is going to be here. Okay? And here, because you've understood the relationship of the operator to the inducer, so to the repressor protein. So in this situation, because the repressor was functional, it means all these positions have got a roadblock. To unblock them, I need lactose to be present in the environment. That lactose must not be in the form of lactose alone, it must be in the form of allolactose. The allolactose will bind to the repressor protein, move it away from the roadblock, and bind to this one also, move it away from the roadblock. When that is done, the promoter now can transcribe the genes that are downstream, but transcribing genes that are only function, not the ones that are not function. That is why in this situation, if no lactose is present, if there's no lactose that is present, the roadblock will still remain there because there's no one to unblock it, meaning you won't get any beta galactosidase. Now, like I said, when there's lactose in the environment, the lactose now can be converted to allolactose. What is allolactose going to do? It's going to unblock the operator. When the operator is unblocked, your genes downstream can be transcribed. Those genes that we're saying can be transcribed are only those that have got a plus sign. You understand, like this one, meaning that what you see here as beta galactosidase being produced when lactose is present is not coming from this gene. Yes, of course, even this operator is going to be unblocked, but the polymerase cannot synthesize a gene that is defective. Okay, it will only synthesize in this. Of course, it can, because the times when the gene has got mutations, it will synthesize it. But we're thinking of this situation as hypothetical, to say if the gene product is not functional, there'll be no transcription. But otherwise, if here there's DNA, it will read whatever is there. Okay, but here, hypothetically, we're trying to say there'll be no gene product if this is a Z. So the gene product we're seeing here is coming only from this copy. Guys, up to this point, is it clear? I can move on to the next one, right?
Yes, sure, madam.
Okay, so let's move on to the next one. Okay, the next slide. Let's look at the second combination. Did we do the first one? I think we did, right? I can't remember, but anyway, let's look at this combination here. So again, like I said, the first thing that we need to do is to understand the relationship between the I gene producing our repressor. You know what to repress means? You know, even in a conversation in a room, you know that those people want to talk too much, repressing what their friends are saying, like repressing someone, you're blocking someone. You know, the people just are repressors. Even in relationships, they're there. You're with your girlfriend, every time she's, let's go here, before you know, she's telling you where to go. If you, it's your boyfriend, every time you want to say, let's go and do this, the ones who are directing, kind of repressing, you know, even the suggestions, the good ideas that you have. So repression in this sense is blocking. So the first thing you do is to understand the relationship between your repressor gene and the operator. Both of them, you must understand whether they're functioning or not functioning. So in this situation here, the second column, we have I minus. Here, I plus. I want to understand what that means. When you say I minus, it means that gene is not working. It's sick, it's defective, it's non-function. What about the same position on the other copy? Like I said, we all carry two combinations of the same gene, alleles. We'll look at them very shortly. Okay, what that means is that this copy, because it is functional in this system, we're considering it as one thing. This guy here is able to produce my repressor protein. And we know when that repressor protein is active in the system, it's going to look for what? Any functional operator. It doesn't matter whether it is on the same gene copy or it's on the other. It just cares to say, I'm looking for any operator who is functional. So at least that we've understood. Let's go to the operator. I've skipped the promoter. I'll go back to it. I want us first to understand the relationship between the repressor protein and the operator, because when the repressor protein is active, what is it looking for in the system? It is looking for the operator region. So I want to understand, at this point, I have established that in my system, I'm going to have a repressor because this gene copy there is active, not from this one because this one is defective. Now, this repressor protein, when it is produced, we said it is acting in trans. It can function both in cis, affect its own gene copy, or it can also affect the other one that was not produced by it. So this repressor protein, once produced, it can bind to its own operator. Why? Because this operator is functional. How do we know? There's a plus sign there, isn't it? Now, what about the other gene copy? This repressor protein is active, and we are saying the repressor binds to the operator. Can it bind to this guy here, operator? It can't, because we can see this is OC. And that means that the operator region is not functioning. It is sick, okay? The binding site cannot bind the repressor molecule. Because of that, these genes that are down this side, like I said, if the roadblock is unblocked, all the genes that are downstream here can be produced, but on one condition, they must also be what? Function. In this situation, I'm trying to understand the relationship between the repressor molecule. At this point, we all know came from this gene copy. At this point, we have a repressor protein diffusing from the kitchen to whatever bedroom you should go to. But who is it looking for? The operator. It has found an active one, so it can bind and it will block until someone comes to take it out. But when it went to this gene copy, it didn't find anywhere to what? To bind, because this operator region is sick, it's defective. Because of that, these genes that are downstream can be transcribed on one conditions, they must be functional. How do you know if they're functional? You can look at the LAC-Z. The LAC-Z is positive, meaning that it can be expressed. What about the LAC? So the LAC-Z is functional, meaning it can be expressed. What about the LAC-Y? The LAC-Y is sick, defective, because we can see it is minus. So while this operator cannot bind anybody and it is free to allow polymerase to go through, we can get our LAC-Z, but not our LAC-Y because this guy is sick. What about this position here? The moment the repressor protein binds to the operator, it creates a roadblock, meaning that these genes here can only be transcribed under certain conditions. Remember, when we say these genes are transcribed under certain conditions, it's important for us to understand the bacteria doesn't want to invest or waste energy in what it doesn't need. So it can only turn on expression of LAC-Z or LAC-Y when they're functioning, when there's a need. When does that need arise? When there's lactose in the system and there's no glucose. It's like right now, if you haven't eaten your breakfast, there's no need for our body to start producing amylase. It's a waste of energy. What is it going to be doing in our mouth, in our saliva? There'll be residue, but what is it going to be doing there? I don't know if it wants to digest my tongue, but I'm not too sure. In my stomach, I should be having pepsin and all those enzymes that digest my proteins. For what? They'll start eating up my stomach. So those enzymes, even my system, needs to conserve energy. They should only, be produced when there is a situation that calls for them. That's how you should understand this system. When you say lactose plus, it means that now the bacteria has a situation where it has been given food in the form of lactose. Because lactose is in its environment, it needs to eat. Now, for it to obtain nutrients from lactose, it needs to express these genes. So there's no need for you to express them when there's no lactose. In addition to that, there's no need for you to express these genes if you've got glucose, because glucose is easy for you to use. Okay, don't spend more energy. It's like food. You've got a choice to just eat food in a liquid form and one which requires you to chew, like a chiwaya, the roasted maize. Chewing that ziwaya is too difficult, it's a lot of energy. In fact, it can even give you a headache. But if you're given very soft food, it will be easy for you, to eat because you don't spend so much energy. So the bacteria chooses glucose because it's easy to utilize, less energy spent compared to this. So this whole system, understand it in the simplest way. These genes are only expressed when a need prevails. The need is no glucose, lactose is present. So let me go back to my scenario. When I don't have lactose, I want to find out whether I'm going to get this gene to be produced. Remember, under normal conditions, they are only produced when there is lactose. So right now you've been told in that system there's no lactose. So I'm going to be answering I'm going to be filling in these boxes one at a time, not looking at both conditions at the same time. I'll just pick one. I want to know whether with this genotype, by genotype, they mean this gene combination. This gene combination means I've got a plus and a plus. So that's one gene combination. I've got P plus and P plus. In fact, I even forgot to mention, when you've got a P plus, the P position there is a promoter, isn't it? The promoter is that position on your DNA where your RNA polymerase will bind. When the RNA polymerase binds to the promoter, what does it do? We hope it can help us transcribe our genes in the process of transcription. In this case, what genes is this RNA polymerase going to transcribe when it binds to the promoter? It is any of these genes. Any of these genes only if they function. In a similar way, when the RNA polymerase binds to the promoter, what we hope it can do is to transcribe any of these genes only when they function. But the polymerase itself, when it binds to the promoter, like in this case, the RNA polymerase can bind to this promoter, can also bind to that promoter. But here, we can see that the RNA polymerase, once it is bound, it's not obstructed by any roadblock. It can freely give us any one of these, okay? But the situation here is different. Even though the promoter can bind the RNA polymerase, there's a policeman here. I hope none of you guys are a policeman. When there's a roadblock, when you're driving on these roads, the moment you see them, You even know they need to stop. So here we can think of it as there are policemen here. Don't quote me, guys. Anyway, what we're saying here is that there's a roadblock here. The roadblock because we have got our repressor molecule bound there, meaning that even though the polymer is binding the promoter very well with your sigma factors and everything else, it cannot give us any of those genes. Which genes are we talking about? Only those that are functional. How do we tell functional from non-functional? We are looking at the position plus or minus. When you say Z minus, it means this gene position is not function. If I go this other side, when you say Z plus, it means this gene product is function, meaning that if there's a situation to synthesize the gene via polymerase, the gene product won't come from here, but at least this bacteria can still survive, utilize lactose because this guy here is functional. And this is how life works. Some of us carry mutations that we don't know about. Not that we carry both functional gene copies. Some of us even have certain gene copies that are not functional, but because we've got another one, we can function normally. Okay. So now let's go back to our situation. No lactose in the environment. When there's no lactose in the environment, what does that happen? The operatable of binding to the operator region. If it can't bind, here there's no roadblock. There's nothing going to stop this polymerase from transcribing these genes. So what it means is that in this regard, whether there's lactose or no lactose in the system, this operator region is free. It's like, if you live in an area where these police people always put roadblocks, and the roadblock is not there, you just freely pass, isn't it? There's nothing to stop you. But then the day you find them there, you have to stop. So think of this situation here as that road that where you normally find these police people, especially those places near these police stations. Okay, think of it to say they're no longer there. So cars will just be passing. So in this regard, there'll be transcription of Z, but lack Y, there'll be nothing, even if the operator is free, because this gene product is sick. So meaning that's why you C plus. What will happen when there's lactose? It will be the same thing. Why? Because this gene copy here, the Z, there's nothing to block it because the operator region is sick. Now, why is this operator here not affecting this one here? This is because the operator itself functions in cis. If this operator is working, it can only block the genes, it can only affect its function, can only affect the transcription of the genes next to it. You can't use it for the other gene, for the other chromosome. That is why this operator here, it functions in cis. The operator functions in cis. What do you mean by that? If this guy here is defective, it does not mean its defectiveness will only affect its own genes downstream. It cannot affect anyone this side. Like in this case, it is sick, meaning that if the operator is sick, it can't bind the repressor molecule even if it is present. So in that they won't get any gene product, at least from this situation. What about this other situation? This other situation needs the presence of lactose for me to get, lac Y. Why? Because lactose penase. Yes, lactose penase. Yes, I'm going to get it because this gene here is positive. Am I going to get it from this side? No. So I'm looking at this position together with that position. That is why you get negative when there's no lactose and positive when there's lactose. Guys, at this point, this is the best I could do. Okay, now I need someone else to attempt just this one so that we can move to the next topic. Is that explanation clear? I think some of these things you might have to, take your time after the class and then go through them, watch some YouTube. And of course, you can still reach out. Sometimes even at the personal level. So now who wants to give this one a try? We're looking at this situation here. You can pick any of them anyway. And I just want to mention, when you have got IS, this is a super repressor. A super repressor, what that means is when it produces a repressor protein, it doesn't care whether there's lactose in the system, there's no lactose in the system, the genes will be turned off. It doesn't care. Just to let you know what this means. When you have IS meaning that the inducer gene, when it produces the repressor molecule, it will remain fixed on the red block. Do not allow for transcription of these genes. But anyway, now, I'm asking you, so we won't pick any of these with an S. So you can pick this one if you want. You can pick this one. You can pick anyone or just pick one. Guys, are we going to sleep or are we going to have lunch? It's 12 hours, I know. You know the problem, and you know the good part about it is next week, no, just very soon on the 3rd of July, I'll be able to see all of you. So this time around, you can sit quietly like this, but you can imagine if it were in class, I would have picked you up. So right now, I don't know whether you're sleeping, you're going to have lunch, the aroma diffused and entered your room, but I need an answer.
So, madam, like when giving the answers, are we supposed to tell you that on like, is it no like Shan, it's positive or negative what?
You're in election, we'll end up. You guys, what are we saying here? So you can start. Which one are you picking? So tell me which one I'm picking. Number what?
The second
Start.
last one, madam.
This one?
Yes,
Right?
the second last one.
Okay, good. So start telling us the laksha and that whatever. So here,
Oh, okay.
are we going to have a plus or a minus? And tell us why. So,
madam, me, I'm just guessing though. I'm
no,
just
there's no
using
guessing.
like my.
Please guess. We are encouraging you to guess because remember, in the test, there'll be no guesswork. Remember, you get a negative, right? So we know there's no guessing here. So at this guess, and one is going to penalize you right now. So please give it your best shot. Okay.
Yes,
So
madam.
we
Like
can start.
the criteria me I want to use, like
Uh-huh.
on that one, is it the replacer, the I plus?
Uh-huh.
So like on the first one we have the I plus, and then on the other one we have the negative I. So
Good.
like on this one we are going to have a positive on no lactose.
How have you come up with that? Please help me understand. I don't know the answer by the way as myself. I also need to go through each one of them stage by stage. But for you, you're saying that if I get you correctly, is it Ndawa? Ndawa, you're
Ndawa.
saying Ndawa. You're saying that because we have I plus on this chromosome, and here we've got the pressure protein.
Yeah,
Yes, right?
because that one is active, yes. That one is active, yes.
Yes. What about this other gene? Are we going to get the repressor molecule?
No.
No, good. So then you and me can agree to say in this system, we're going to have a repressor protein. Now, remember what I said about the repressor protein. You, if you have a girlfriend right now in Dawa, if your girlfriend, you know, represses you, suppresses you, it means that you can't make any decision, right? So you understand
Yes.
what I mean? You have a girlfriend.
Yes. No.
I hope she didn't hear you. Okay, now, guys, what we're saying here, I'm telling you, what we're trying to say here, and I take that back. I hope she didn't hear anything if she's there. What we're trying to say here, guys, is what Ndawa is saying is that because the I gene here is functional, we are going to have our repressor protein. What is the repressor doing? It's going to repress the transcription of the genes that are downstream, isn't it? That's what you're saying, Ndawa, isn't it? But I want to
Yes,
understand.
madam.
So when you say here, we're looking at this system here, there is no lactose in the system. So should I put a plus? When you put a plus, it means that you're going to have transcription, right? When you put a negative sign, it means there's nothing there. It's like a zero. That's what this plus and things, the minus means that you want to get that gene, the gene product. The plus here, it means you're going to get beta galactosidase. When you get a minus sign here again, the minus means you're not going to get your lactose. The plus sign means you're going to get the lactose. That's what these things mean. Remember in the previous one, you had the whole, you know, enzyme written to you, but this is not necessary. You can actually simplify it by just putting plus or minus. Plus to say it to be present, minus to say it to be absent. So Ndawa is saying that in this regard here, it should be plus. How many of us agree? And why? Why did you say that? Of course, he's given me his reasoning to say because here it is plus, here it is minus, here it will be a plus.
I think I made a mistake. It's
Good.
supposed to be a negative.
Okay, why?
So here, the lactose, it's going to be present. So we're going to have a negative on the first one, and then on the plus, that one, we're going to have a positive.
Guys, I want someone to record what's what's in there. So we're going to have plus no lactose, it will be negative. On the plus lac, it will be positive, right?
Yes, madam.
Okay, good. I don't know whether that's the answer, but can someone else give it a try? As in, we want to work through why you said that. But now, how did you get to that answer?
Madam, I'm just using the guessing work.
Now that's
Like
dangerous.
on the first one, I'm using the first one as my reference.
This I plus and no, but it can't work like that. Remember, I think I've run you through how to come up with the potentially correct answer. It could also be wrong. You know, when you're looking at these things, sometimes even myself, I could be wrong. But I'm hoping that as I go through, you're also able to fall. So at this point, there is no way you're just going to look at this position alone and not consider everything down there and say it's going to be positive or negative. I just want to emphasize one point. The same way that I was walking through stage by step is seemingly the same way you're going to get to the answer yourself. So just by looking at one position, for example, I can't just look at the position Y here, negative, Y there, positive, and say negative here, positive there. No, I need to consider this. Remember, it acts, we said these are operons, isn't it? It's a cluster of genes that are going to be transcribed together. So meaning that you must look at them as a whole, not individually. Okay. So I need someone else to agree with what Ndawa is saying. And you know the times when you guess and the answer is correct, huh? But I don't encourage you to do that. It's dangerous.
Madam, can I try to understand on the I plus part?
Go ahead. What do you mean? You want to explain what you've understood?
Yes, like from what you've said, when we're looking at the I, when the I there, the first letter
Okay.
is a plus, meaning the
Okay.
replacer is active.
Good.
So it, what I can say is, it more like creates like a roadblock to stop the RNA polymerase from moving forward, meaning transcription will be turned off.
Good.
Yeah, so that's what I understand. So if it's a positive, I can say when we're doing the no luck thing, it has to be a negative.
So if I is that shepherd? Okay. Now
Yes.
let me just recall what shepherd has said, or let me just try to understand what he's trying to say. So here, guys, we can see this is I plus, right?